And we are back for the last anticipatory briefing of the day. We are going to welcome someone who's the director and professor of medical mycology. She's in charge of National Reference Laboratory of Antimicrobial Resistance in Fungal Pathogens at the Department of Microbiology of the Wallabhai Patel Chest Institute at the University of Delhi. I said that right because it's spelled Wallabhai, W-A-L-L-A-B-H-B-H, double B-H. Find another word with a double B-H. I've never seen that. She works on molecular ecology and population genetics of pathogenic fungi, antifungal drug resistance, and currently she focuses on Candida auris, drug-resistant dermatophytosis, and rare yeast infection. I think you have some explaining to do, some of these words. So we're going to have a slightly longer presentation than on the other sessions, but we agreed on 25 minutes. Still gives us a lot of time to take questions. You're going to tell us about fungal pandemics. Please help me welcome Anuradha Chaudhary.
Thank you. Thank you very much. And I'm really, really delighted to be here. And I'm going to take you to the journey of fungi, fungal kingdom world, and how fascinating it is. So my first slide will be a little bit of fungi which cause human infections. But before I start with these kind of infections, I would like to tell that how the fungal kingdom is so diverse, what all good things the fungal kingdom has. So we all know we encounter fungi, which are millions of fungi on the planet. And what common fungus you have seen with your naked eye, someone can say, we eat that fungus, and when we're walking in the woods, we see mushrooms. That's a fungal pathogen. They're not pathogen, that's a fungi, right? But what, as a clinical microbiologist, as clinical mycologist, I see under the microscope are the fungi which infect humans. And there are a range of fungi which infect humans, but very few of them are characterized. Not only that, fungi are very helpful. They support life on the planet. And how do they support life on the planet? They are degraders. So they degrade the nutrients from the dead plants and animals, and they recycle the energy. So they're beneficial. They support life on the planet. And also a very important aspect of fungi, which we don't know, is that they are very important because they produce very, very beneficial antibiotics which we use to treat our patients for bacterial infections. And also immunosuppressive drugs which we give to the patients who have organ transplantation to prevent the rejection. So many of these immunosuppressive drugs are derived from fungi. So fungi can be friends. But now I'm going to talk about the fungi, how much threat they're posing to human health, how much threat they're posing to our wildlife, and how they are threatening the crop production. So if you look at these slides, as I said, that fungal kingdom is very, very variable. Similarly is the infections which they do. So in immunocompetent patients who do not have had compromised immunity, they generally get skin infections. So these skin infections generally, it comes in mind as like worm, tinea infections, which can be treated by a simple over-the-counter drug. But this is not happening now. So if you look at these skin infections, 1 to 6 billion people on the planet are affected by skin infections. And these skin infections, the population is about 80% of the planet suffer from this disease. And also it is the 4th most common disease after dental caries and headache caused by fungi. And secondly, if you look at the tongue and you see the white plaques, this is a typical tongue where you give maximum antibiotics to patients, and then this candida, this mucosal infections occur. It also occurs in bottle-fed infants, in preterm infants, and these infections contribute about more than 100 million recurrent cases of mucosal infections occurs throughout the globe. And also, women significantly suffer from candida infections, vulvovaginal candidiasis. And these vaginal infections, they affect about more than 300 million women over their lifetime. So this is the burden of fungal infections when we see. And not only that, skin and mucosal infections, but we have a lot of allergies. We have chronic fungal infections when we have change of weather, because fungi are present in the spores are present in the environment. And quickly to show you the scenario, if you look at these slides, they can be very, very disfiguring. Such kind of invasive infections, if you see, they happen in AIDS patients. The first, on the extreme left side, these are the nodules of disseminated infections of endemic fungus, which occurs in Southeast Asia. Then you see a neonate, a preterm newborn, and with invasive devices, they get bloodstream candida infections in intensive care units. Then you see brain infections, very, very high mortality. And when you see such a disfiguring figure, this is one of the cases where you just get the infection by trauma, and then it disseminates to the entire body, and then you do not have antifungal drugs to treat them. So if you look at the second slide is to convince you that although we have invasive fungal infections are the ones which we are dealing, which has very, very high mortality. And if you look at this mortality, untreated mortality, it is more than 90%. The patients will die with these fungal infections. And these 4 important fungal infections, which I have on the left side, like aspergillosis, candidiasis, they all occur in our hospitals. And if you look at the treated mortality, even if you have antifungals, you treat them, the mortality is high, about 50%. So what is the problem with these infections? Why can't we treat them? So when you look at the figure, 1.5 million people die each year with these fungal infections. And it is one of the 5th largest cause of death. And what is more important is this annual mortality rate from fungal diseases. They greatly exceeds than that of malaria. We have enough funding for malaria. We talk about malaria, but fungal infections, people die more with malaria, with fungal infections than malaria. And it is same as tuberculosis. People who are coming from countries where tuberculosis is endemic understand that tuberculosis has a huge load. But look at fungal infections, under-recognized. And we do not have enough surveillance data, but if you look at from the United States perspective, $5 to $7 billion, it's a healthcare cost due to fungal infections. And we do not have data from other parts of the world. And these infections, why they're misdiagnosed? Because we do not have capacities in the lab. We have a limited access to the diagnostics, and we do not have good, effective mycology labs in all the parts of the world. It's limited to certain part of the world, but not Latin America, Africa, Asia. We are equipped well to diagnose these fungal infections. So the most important cause of death of fungal infection is underdiagnosis. And what has helped, the WHO has just recently, in 2022, end of the year of 2022, have released the first ever list of fungal pathogens. Before that, we did not have any recognitions. And they have given our priority pathogen list. They have categorized certain fungi in critical group, in high group, and medium group, depending on the antifungal resistance, the treatment protocols, and also depending on the burden of the fungal infections data, which is missing from major part of the world. And if you look at the annual mortality due to fungal infections, there are 4 important causes, which is Cryptococcus, which cause meningitis, and specifically in AIDS patient, and also Candida, Aspergillus. These are the group of pathogens which cause 90% of the disease. And this list is a real game changer, because now it has given us, it is a guide now for future research, for future policymakers' interventions. So I think the next 10 years, it will really serve as an important guide for what to do about fungal infections. And I'm going to quickly take you at what fungal pandemics recent years we have faced. And this is one of the most important, Candida auris. Auris means from ear. So this was a harmless pathogen labeled in 2009 from Japan, from the external ear canal, sitting quietly, not causing any infections. But then what happened? It has become a global pathogen. Less than 10 years. In next slide, I'll show you. It has gone more than 60 countries. And why is it important? Because we have only 3 or 4 classes of antifungal drugs, not like bacteria, hundreds of antibiotics. And if this pathogen was the first one, which was multi-drug resistant, it started with resistant to 2 classes of drugs. So what do you treat? How do you treat this pathogen? And then it has an ability to survive in the hospital environment, in the ICUs. It can be transmitted from one person to another person, and once these patients have devices, it goes into the bloodstream. It causes infections, which has a mortality very high, 30 to 60%. And on the top of it, we do not have drugs because it is multi-drug resistant. So this drug-resistant auris, we also studied at the time when we had SARS-CoV, and we saw that in our ICUs, those which were severely sick patients had a great high transmission of Candida auris, and the mortality was also high because of COVID and also because of Candida. And this is the slide just to show you that it began in 2009, and the first, initially, there were no invasive cases. Only in Korea in 2011, 3 cases were identified of bloodstream invasive infections. Out of those 3 cases, 2 died. But then we have the biggest outbreak at that time in India. We reported a huge outbreak in 2 our hospitals and said, look, it's a problem. It's not going to stay in India. It's going to go global. And actually it happened. Now if you look at it, 2022, 23, more than 60 countries, 4 continents have this pathogen, reporting huge outbreaks in our ICUs. And it is a huge problem now in USA. And if you look at this paper from CDC, they clearly say is that only in 2016, the first case came. And then now, in this paper from CDC, they say that, okay, we have a 44% increase in 2019 cases, invasive cases of auris, and 95% increase in 2021. So this is the scenario. And more troubling is that the drug resistance, echinocandin is the drug of choice for this pathogen. And that too is becoming resistant. So they're experiencing resistance, which is increasing 3 times in each of the previous 2 years. So the climate change has a huge impact on fungal infections. You know, the warming climate and the global warming, this pathogen probably has emerged because of the high heat, because this can thrive at 40 degrees Celsius. This pathogen can grow at 42 degrees Celsius, which is unusual of several fungi, because they are saprotrophs, they grow at lower temperature. It has adapted to higher temperature. And this was the hypothesis which was proposed by a very important scientist. And they said probably Candida auris is emerged because it grows at high temperature, because it tolerates salt. So probably it existed in wetlands as a very quiet pathogen. And then due to warming climate change, it has adapted to higher temperature. And by the human activities, we have got this pathogen in our system. So we worked on this hypothesis. And then we chose Andaman Islands in India, where we have all these scenarios, where we have wetlands, where we have salt marsh, where we have these islands which are not open to the human activity. So we collected the soil samples, we collected the water samples from these beaches and from these areas, and what we observed, yes, the Candida auris was there in these 2 locations. And one of the location was salt marsh, and with restricted human activity. And what we observed quickly, I want to show you, that isolate was susceptible. If you look at the 16-degree concentration, it was inhibited by antifungal. So probably it existed as a susceptible isolate. And also, if you look at the graph of the growth pattern, so if you look at the orange color graph, it is growing slowly at 37 and 42. So probably this was the hypothesis, which is proven by this study, that yes, Candida auris existed in our environment, in the coastal wetlands, and a lot of changes which are happening because of use of antifungals in our environment as well as in our hospitals has made this pathogen resistant to antifungals. And here I would like to take your attention that dual use of antifungals in the crop production, material preservation, and also in human health is causing a lot of stress on the fungi. So we have azole drug class of drugs, which is very important for several part of the world because it is cheaper as compared to other drugs. And these azoles, which we use for human, are also used in the agriculture setups to prevent plant diseases, crop diseases due to fungi. They have a similar structure. And the continued presence and usage of these drugs in the agriculture environment has made these pathogens resistant to antifungals. And how do we get this infection? Because the fungi, as they stay in our environment, we inhale them. So naturally, immunocompetent patient will not have invasive disease, but a lot of people, due to organ transplantation, steroids, so many immunity disorders, when they inhale this fungus, which is already resistant to the antifungal, we have no choice to treat them. So the dual use of azoles in the environment and the hospital is concerning. And this is the depiction that one of the example is Aspergillus fumigatus. It has a very high mortality. So this fumigatus is present in our environment, and it's not a plant pathogen. So, but when we spray these fungicides in the environment, which has a similar structure, so they select the resistant isolates, and when we inhale, and specifically patients who are immunocompromised, when they inhale, they have this infection. So it's the same mode of action as agriculture fungicide, the same mode of action as clinical antifungals, has actually resulted in it has resulted in acquiring resistant isolates from the environment. And quickly, I will tell you that also, the fungicides are used for storage of fruits, and we just did a study. We wanted to see the morphology, diversity of fungi on the surfaces of fruits, and what we observed that fruits which were stored, which had more fungicides, they had presence of Candida auris, as compared to the fruits which were not exposed to fungicides, which were freshly picked up. So, and quickly, also I would like to say that these strains are quite similar to the strains genetically which infect humans. So these strains are very similar. So what's happening in the environment is what we are getting. Why I'm telling you that this is because it's connected. It can be transmitted. The transmission cycle can be there if we use this. The fungicides which are used in the environment can also select resistant strains in the environment, which can be potentially transmitted to humans or vice versa. So this is how, what we are doing is we are putting a lot of pressure of drugs in the environment, and the colonies, fungal communities are changing, and those fungal communities are in turn coming to our hospitals, to our environment. So now I shift your focus to second pandemic. And this is very, very simple. We know that whenever we have this itching, we have this small kind of worm, tinea infection, we get this over-the-counter availability of antifungal creams, we put it and say, okay, we are fine. But what's happening in more than a decade using these creams, this kind of scenario, now we see this kind of fungal infections difficult to treat. They're resistant to the first class of antifungal, terbinafine, which used to be very, very effective in treating these cases. And this strain has not been limited to one particular country. So what we did initially, this trichophyton species which we found out in 2016 to 19 in India, which was resistant to the terbinafine, the commonly used drug due to mutation in one of these genes, we found that this has not. Let me show you this here. So we said that, this strain is not what it was previously, either interdigital or mentagrophytes. By genome sequencing, we said, look, it is something different than those strains, but it's rapidly spreading the resistant alleles. And this strain has now spread to more than 30 countries. And it was given a new name, Trigophyton indotinea, which is a newly, highly terbinafine-resistant dermatophyte species by the Japanese group of workers. And it has now, at present, spread to several parts. And it is very, very contagious. If one family member gets it, another family member gets it. And then there's a transmission in community happening. We are traveling, we are taking our fungi with us, we are also taking our creams with us. So this is the scenario, what we are doing. And also, quickly, I want to talk about the I don't know whether a lot of people would have heard about black fungus and mucormycosis outbreak. When we were having the COVID-19 in India, we had this huge outbreak of mucormycosis. But it was not only in India. Several parts of the world was also reporting the cases, but we had lots and lots of cases of mucormycosis at that time. Why? Because we have a large number of diabetic population. And also, the steroids were given in excessive doses to those patients. And once we had this imbalance scenario, black fungus, inappropriate to call black fungus because the fungus is not black, but the media was projecting it as a black fungus. But patients' presence with this black eschar, it affects the brain, it affects the eyes, it affects the sinuses. So even if the patient is treated for SARS-CoV, but this was what was happening. And the problem was misdiagnosis, underdiagnosis. So when you do not know what you are dealing with, you all the time gives more antibiotics. And when you give more antibiotics, you also pose threat to the bacterial population. So they also get drug resistant. So this is a scenario when we are not equipped to diagnose fungal infections. So what we are creating is another problem, creating resistance in our bacteria also. Poor recognition, misdiagnosis, antibiotic overuse, all this can be prevented by accurately diagnosing fungal infections. So what can we do? So first of all, there is a limited access. There is a limited access to essential diagnostic tools to identify these fungal infections in Latin America, in Asia, and several parts of the world. Even the well-equipped laboratories in North America are not equipped for doing the susceptibility testing. And that is very important to know what antifungal resistance is happening. So this disparity has to go. We have to have an infrastructure. We have to have mycology diagnostic laboratory capacities everywhere, everywhere. Because the fungi are not going to stay in one particular country. You have seen 3 examples, how it started from one country and how it has gone everywhere. And the most important limitation that we cannot convince the authorities is because we do not have resistant data. We do not have, because surveillance is limited. It's not a notifiable disease. We do not do susceptibility testing. So if we have to address this threat of antifungal resistance fungi, we need to have the surveillance data from different parts of the world. Something which is resistant to Asia may not be resistant anywhere else. So the drug usage policy will be different. So very, very basic data, which we already think we have for bacteria, we do not have in 22, 23 still for these fungal infections. So what can we do? I mean, advancing our laboratory capacity. That's what I like to do in the next 5 years. I mean, advance our laboratory capacity. Make them capable of diagnosing fungal infections so that they can properly diagnose and decrease in these empiric treatment. Oh, it may be fungal infection. Let's give 2 or 3 antifungal drugs. That can be prevented. And of course, WHO, when they released this fungal priority pathogen list last year, but in 2019, they already prepared a protocol for inclusion of Candida species from bloodstream isolates. So these are the positive things which has happened. And inclusion of Candida species in glass, in glass surveillance has helped us making the infrastructure of many countries for the fungi affordable, feasible. And priority pathogen list, which is released, I'm sure it will serve us as a guide for future. In next 10 years, we will be working on these important priority pathogens. And also the policymakers will take into view that these fungi, these are the ones which we need to work because these are the ones which affect globally. And also I want to draw your attention here that we have only very limited class of antifungals because the fungal pipelines, the fungal drugs, they take 20 years, they take billions of dollars to develop. And just recently, we have been very, very fortunate that promising new compounds have come out for human use and not, phase 2, phase 3 trials. And also, these two or three important drugs which have come up, these are the ones. Among them, alorifim, which is very important for invasive diseases, it has the same mechanism of action which a recently authorized fungicide has. So if we do not have a One Health priority to look into the same dual use of antifungals in the environment and in the humans, we will lose the potency of these new drugs also. So we need to prioritize One Health approach in formulating the policies for usage of these drugs in both the situations. Yeah, that's what I want to say. And also, I'd like to take your attention that with SARS-CoV, we had vaccine development so fast. We do not have any vaccine for fungal infections. We do have experimental data, we do have for some important fungi, but none is commercialized. So in next 20 years, 15 years, I also want to see that we have these vaccines available, for fungi. Now, quickly coming to the another aspect of fungi. So how fungi are threatening our food security. So first, being a clinical microbiologist, I showed the spectrum of fungi which infect humans, but what they are doing to our food security in last few years. So both essential calorie crops, wheat, rice, and also our coffee, bananas, all of these crops are afflicted by fungal diseases. So that is why a lot of medicines are used in agriculture to prevent these crops, from fungal diseases, because then they will jeopardize the food security on this planet. And one of the examples, the rice blast disease is caused by a fungi, which affect about 12 to 30% of our annual rice harvests. And also, not only that, wheat can be affected by our fungal infections, and it jeopardizes our harvest from Europe, Asia, America, and also bananas, monocultures. They are completely decimated by Fusarium. Fusarium is a fungus which infects humans also. It also infects our banana monocultures. And we were talking about mycotoxins. Yes, absolutely correct. Mycotoxins are a threat to human health. And these mycotoxins, which are secondary metabolite of fungi, they actually contaminate our 60 to 80% of crops post-harvest. And what do they do to humans? I mean, if they are eaten, if they are consumed, so they cause life-threatening liver damages and also implicated for cancer, right? So this is what fungi are doing to our crops. And very quickly, I also want to bring the attention to the animal kingdom. Our ecosystem is changing. We are threatening our ecosystem balance. And because the fungi are also affecting our amphibian population. So if you look at chytridomycosis, it is really decimating the amphibian population. It's declining and causing the extinction of amphibians worldwide. although the research is more focused in some particular areas of North America, Europe, but actually it is infecting the entire population. And that is going to alter our ecosystem because these amphibians are important for our food chain security. They're going to eat those pests. And if the balance is gone, I mean, we are nowhere. And secondly, if you look at the white nose syndrome in bats, this fungus is really devastating. It's killing the entire colonies of North American bats. And so we have to take care. We have to see how we can prevent this. And I'm not going to talk this, but I'm going to show this. So I don't know how many of you have played this video game or seen this HBO show. And the zombie fungus, actually exaggerating. No, it's not going to kill the vertebrates. It does not infect that. But the show is a reminder to all of us that if we do not take action at a global level, fungi are there for us. They are our friends, but they're going to kill us. We have to live in harmony. We have to have a planet where our fungi live with us helpfully. We know that fungi are very, very important for food stuff. We love our wine, we love our beer, we love our cheese. All these are derived from fungi, right? So let's have some concentrated efforts to deal our fungal infections. And I'm thankful to Vallabhbhai Patel Chest Institute, what you're saying. So we run the National Efforts Laboratory. And here, it's my duty to say that Canadian Institute for Advanced Research is the first platform which has brought mycologists together. And actually, that platform is important for the mycology community. They have brought plant mycologists, they've brought human health people, and they are actually promoting the cause of fungal infections. So we have a very exciting director. We have a Fungal Kingdom group there, and we have a very exciting director, Joe Heitman and Lee Coven, and Danielle is here. Thank you very much for your attention. I hope I'm in time. I took more.
I wish that slide was still on. People could see the Valab by BHBH. So please send in your questions through the app for me to relay. I think it was quite an intense presentation, I must say.
Yes.
Some of the pictures were hard to look at.
Yeah.
It's quite dramatic.
So you're scared?
I'm scared. For me, fungi was like
Friends.
Roquefort, truffle, bread and cheese, and
That's what I said in the end.
All kinds of really cool stuff. And now I realize it's kind of a silent killer that's coming for me. So I won The Last of Us. I killed it, but it was a video game.
Yeah, okay.
So it's really coming. I cannot do what that kid does in the game, in the movie. I'm going to go straight to the questions. What do you think we can learn from other research and policy fields, such as antimicrobial resistance and pandemic preparedness? So that question already existed with microbes, viruses, what can we derive from these past wars for the upcoming fungus doomsday?
Yeah. So for antifungal resistance, it can happen when we give long-term antifungals to the chronic diseases, like when we have this class of drugs, when we give them continuously, so that cause mutations in their genomes, and that drug becomes resistant. So we cannot help it. I mean, that happens over the long-term usage in human health. But what I'm also worried is when we have the similar drug in the environment, and there the pressure occurs in an innocent bystander fungus, which is not causing any disease, but then that fungus is becoming resistant, and then we are acquiring it, and we already have a resistant pathogen where we cannot treat. So what can we do? We have to have certain policies to detect that kind of resistance pattern which is happening in human health as well as in the agriculture, and restriction or policies that how much we can use it, how much we can do, that has to be decided together. I mean, that has to be have in a policy in One Health. We cannot escape that. Right now, if you ask me, we do not have any policies where we cover that, okay, the drugs which are used in clinical medicine are also used because of the food security. We cannot immediately stop that. But yes, that policy has to be driven, has to be made, and that is a difference in fungi as compared to bacteria. I mean, that's there.
Somebody wants to be even more scared, I think. The question is, how many fungi species are estimated to exist?
Millions.
There you go.
Millions of fungi.
Thank you for the question.
But only hundreds of them are pathogenic to humans. But that is not going to happen because I'm not showing here, but recently we saw a kind of fungus which was never reported before. So fungi are always there and they're upcoming, but at present we have about hundreds of fungi which are pathogenic to humans, but the environment are millions of fungi. Millions.
How do they compare with viruses from a detrimental / beneficial perspective?
So that is the problem, Eukaryotic fungi, they share a lot of genome with what humans have, right? They're very close to humans as compared to bacteria. And that is the problem when we design the drug targets. It is difficult because they're very close to humans, so those targets are overlapping, so we cannot have very easy drugs available. And also similar is for vaccines, so they are more close to humans as compared to viruses and bacteria.
Question number 3, psoriasis. Any connection with fungal infections?
Of what?
Psoriasis.
Psoriasis.
Yeah.
Oh, no.
Any connection with fungal infections?
Oh, I know psoriasis because my husband is a dermatologist. Thank you. Otherwise, I would thinking, okay. So psoriasis is a dry skin condition, genetic changes. No, but the fungi. If you give a lot of immunosuppressive drugs in psoriasis, you can have skin infections, but no connection otherwise.
Maybe you could have married a chef. Then that person would be like killing mushrooms as their job. Kind of
and I would not have been standing here and talking about.
Question number 4, is there a connection between microbiome changes and the onset of a fungal infection?
Yes. When we give, a lot of I can give example of yeast infections. So they are there in our flora. They are the part of our microbial flora. So what happened when we give unnecessary antibiotics? So the bacteria which is beneficial is removed, the balance is gone, and the yeast grow higher. And then this yeast cause infections. So if you give, even in ICUs, more than 10 days, 14 days of antibiotics, you remove all your bacterial flora, and then you give a chance for fungi to grow, and then the fungi invade. So yes, there is a huge connection. And that is why, because fungal infections look very, very similar, the initial presentation clinically, they actually happens in comorbid situations. Like the patient is already admitted with some other diseases, viral diseases, or some abdominal surgery or something. So when fungal infections occur, they are not very specific. You cannot pinpoint, oh, this has happened, so this is fungal infection. So they look very similar. The symptoms are very similar as bacteria or viruses. So that is why the emphasis is in diagnosis. If you diagnose, then you will not give excess of antibiotics. You don't give excess of antibiotics, you do not deal with fungi, fungal infections. So all is interconnected. Yeah.
There's a question about vaccines. Could vaccines help with these issues? And the question is which one would you develop if you had a magic wand? What would you focus on?
Yeah. So the vaccines are for cocci, they have for animals in US, right? But human vaccines are not there. But the vaccines which are important are the top 3, Candida, Aspergillus, and Cryptococcus. Those are the top 3 candidates because the 90% of the infections are because of these 3 pathogens. So the vaccines has to be worked for them. Yeah.
Question number 6, is there a way to prevent infections through new behaviors?
Through new behavior.
Through new behaviors. Like can we teach people to behave differently to diminish potential infections?
Yeah. Okay. Very intelligent question. Same. Fungal infections, suppose we are talking about skin fungal infections, right? Skin fungal infections occurs in healthy people, moist conditions. So the behavior changes I will say is when you have a small patch of fungal infections, suspected fungal infections, do not use over-the-counter drug, because they have a very minimal formulation of antifungals. And those minimal formulation of antifungals are mixed with steroids, are mixed with everything. That gives a pressure because the dose is inadequate, right? So that fungus which is there, which is not resistant, over the time, when you keep on applying them for short duration, they remove, and then again you have a itch, you apply it. I'm just giving an example, right? So then you are creating a resistance on your skin. And you are creating a resistance here which is not going to stay here. It's going to go in your family, it's going to go to others. So the behavioral changes is, yes, go to your doctor, find out, have good diagnosis. This is one of the examples where skin fungal infections we are dealing. Now, coming to another scenario like intensive care units. The patient is admitted with, suppose kidney transplant, suppose nephrotic syndrome, for anything in ICU. But suppose Candida auris is there in the ICU. It is there on the bed, it's there on the sheets, it's everywhere. So we have to use infection prevention control practices. We have to teach our clinicians to be very effective infection prevention control practices so that from the environment, we do not transfer it to the patients. And from these patients, it does not go inside and cause the bloodstream infections. So these are the examples where in the hospital level what we can do and at the community level what we can do. I mean, just examples, yeah.
So actually, I'm going to bundle 2 questions. So one was like, why are we behind? Which is the first question I got, but somebody else asked the same question. Why historically have fungal infections been ignored when bacterial infections have had so much investment? What does that mean for developing strategies for tackling in the future? So.
Yeah. So fungal infections were not recognized. Why? Good question. Because fungal expertise is not available. I mean, we have very few people who are working in the field of fungal infections, right? We have reduced funding as compared to others. We do not still have data of the true global burden data from several parts of the world. What I have shown is also extrapolated, published from some countries, calculated. So not a true burden we know. So when we do not know the true burden, how we can convince people that these are important. So also the infrastructure for diagnosing these fungal infections, suppose in a microbiology hospital, in a microbiology lab, you will never find a section of mycology separately. It is always inculcated along with bacteria. So, they're just neglected. They take some time to grow. They do not grow so fast. So we have to include certain specific fungal media, which is not done in routine practice. So let me talk about endemic mycosis, like histoplasmosis and all. They grow after 2 weeks, if we are using culture. So we do not have a system, diagnostics assay, which are very, very rapid to diagnose them. So for over the years, no investment has gone, fungal communities were isolated, and the true burden was not there. So that's why probably they never got a chance to come to the forefront.
Maybe they never saw your presentation. That's why. Sophie, we did question 8 actually, and question 10, there is a word that I've never seen. So either it's a typo or I'm tired, but it's the word phages, Phages.
What?
That's okay. So that's so it's the 9th session I'm doing, and like many times I passed some question from a super powerful brain through me would access another super powerful brain, and I had no idea what I was saying. So this is what's going to happen now. Similar to bacterial antibiotic resistance, where it seems
Yeah, it's working.
phages could be a solution. Are there other topical solutions to fungi based on viruses or other fungi? So who knows what a phage is?
Yeah.
Okay, so maybe you need to, congratulations if you do, but maybe explain what a phage is, please.
I know they're microviruses. I know that, but not much research into these phages at present. Yeah.
So it's not helpful.
Yeah, it's not something which is people are trying or doing a lot of research, but I know these are approaches for fungi which can be done, can be taken. Yeah.
Okay, so we'll have to look up what a phage is after.
Yeah, at present, if you ask me, no.
Okay. We have many questions.
Okay.
Sorry, two more, and then I let you go. Are there any natural medicines that can help control the skin infections before they get too severe? And if yes, do they create resistance?
Yeah.
There's some
Good
kind of magic
question.
grandma trick that you heard somewhere, like put some lemon juice on it. And
I mean, where are the trials? I mean, where are the results? I cannot say anything which is not scientifically proven or done. I'm coming from India, so I know that we always have small things to apply, coconut oil, dry this, that. But I cannot say this in here in the forum because we do not.
you see it, but at a different price.
Yeah, that's a good approach. I mean, try.
You know, we have people in Switzerland. You call them, and they like stop the burns and stuff like that. So some pretty.
We put turmeric.
yeah,
I mean,
pretty
it works. It
pretty
works.
cool stuff.
Yeah, but I'm not coding that. Here. Yeah. Please don't do that.
So last question: could fungi travel on plastic waste in the ocean, thus affecting marine life?
Yeah, but first I'm going to talk about the beneficial thing fungi can do. They are the plastic degraders also. We have found fungi which are degrading this. So they are answer, if we can make them friends rather than just making them foes, enemies.
Let's make that the final word of the session.
That is the final word.
Let's conclude. Thank you very much, Anuradha Chaudhary.
Thank you.
Have a great evening, everyone. Thank you so much.
And thank you, GESDA, for giving this opportunity for fungal world to be represented. I'm really thankful because I really feel that next few years we can do a lot to prevent these infections. And thank you.